This slide shows one example of how the release of a glial transmitter can affect neuronal excitability. In this case, this is in the retina, and it shows that glial cells can both excite and inhibit spike activity of retinal ganglion cells. So in this experiment, these experiments were done in the isolated whole mount retina. The retina was removed from the eye, placed in a dish. The spiking activity of retinal ganglion cells were recorded with extracellular microelectrodes that could record the spiking of the cell. And then the glial cell, in this case, this is a Mueller cell, a radial glial cell in the retina, which functions as the astrocyte of the retina, was stimulated, and the figures on the right show the activity of the neuron following stimulation of the glial cells. So the top trace in each record shows the average spike activity of the neuron. The bottom record shows the calcium level within the glial cell, the Mueller cell, and the arrow indicates when the Mueller cell was stimulated. So the retina is continuously stimulated with a flickering light. That's shown in the bottom trace here. So flashes of light, and for each flash of light there is a burst of spikes in the neuron, in the retinal ganglion cell. And then when the glial cell is stimulated, this causes an increase in calcium in the glial cell, and shortly after this occurs, within a few seconds, there is an increase in this particular cell. There is an increase in the spiking of the neuron. So with stimulation of the glial cell, there is a increase in excitability of the neuron. In another retinal ganglion cell, we see just the opposite. This is the ongoing activity generated by the flashing light, and then with activation of the glial cell and an increase in calcium, there's actually a depression of spiking in the neuron. There is a decrease in the average frequency of firing. Now, additional experiments showed that the glial modulation of ganglion cell activity occurs by the release of ATP from the glial cell. So ATP is a gliotransmitter. It's released from the glial cells, and once ATP is released into extracellular space, it's rapidly converted into adenosine. There are enzymes that break up ATP in about 200 milliseconds or so into adenosine, and then the adenosine stimulates adenosine receptors on the retinal ganglion cell, leading to the opening of potassium channels and to a hyperpolarization of the cell, and this leads to this reduction in spiking. We don't actually know the mechanism for the increase in excitability that's seen in some of the retinal ganglion cells. A second example of glial cell modulation of neuronal activity and synaptic transmission, in this case, is shown in this figure. These experiments were done on hippocampal brain slices, and astrocytes were stimulated, in this case, with an electrical stimulus, and pyramidal neurons were monitored with a patch electrode shown here. So in hippocampal slices, there are inhibitory interneurons which synapse on pyramidal cells, and just as in other synapses, there are spontaneous miniature events that occur. And in fact, if you record from a pyramidal neuron, you can record miniature IPSPs, inhibitory postsynaptic currents, that derive from vesicle release, quantal release of inhibitory transmitter from the interneuron onto the pyramidal cell. The observation that was made in this experiment is that when astrocytes are stimulated with an electrical stimulus, and here's the stimulus shown here, then the frequency of the miniature IPSP events that are recorded in the pyramidal neuron increase. So the glial cell was stimulated here, and after a few seconds we see that the frequency of the miniature events increases. This is at rest, and this is following stimulation. You see there's a much higher frequency of miniature iPSCs. And it was demonstrated that glial cells modulated synaptic transmission in the following way. When the glial cells were stimulated with an electrical stimulus, this caused glutamate to be released from the astrocytes. The glutamate then stimulated glutamate receptors, NMDA receptors, in fact, on the presynaptic terminals of the inhibitory synapses on the pyramidal neuron, and the stimulation, the activation of the NMDA receptors on the presynaptic terminals led to an increase in the frequency of miniature events that were recorded in the pyramidal neuron. So again, the glial modulation of neuronal activity is caused by the release of a gliotransmitter. In this case, it's glutamate, not ATP, and in this case, it modulates synaptic transmission. A third example of glial cell modulation of neuronal activity is shown in this slide. These experiments were done once again in hippocampal brain slices, and in this case the Schaefer collateral axons were stimulated and field EPSPs were recorded from pyramidal neurons. So we're looking at the synaptic potentials generated from CA3 to CA1 neurons in the hippocampus. What's shown in this graph here, if we look at the wild type first, is that as we increase the stimulus voltage activating the Schaefer collateral axons, the field EPSP grows larger. So this gives us a sense of the relation between the number of axons that are stimulated and the size of the synaptic potentials that are generated in pyramidal neurons. So these experiments were done in mice, and the filled circles show the relation between stimulus strength and synaptic potential amplitude for wild type. A second set of experiments were done in transgenic mice, and these were mice that expressed a dominant negative snare domain. This is a domain of a synaptobrevin 2 molecule which blocks exocytosis. And these mice were designed so that this dominant negative snare domain were expressed only in astrocytes. And the effect of this is to block synaptic release by exocytosis, but only from astrocytes. And when the same experiment was done in the transgenic animal expressing the DN snare molecule in astrocytes, we see that for a given voltage, the field potential, the synaptic potential recorded from pyramidal neurons is just about twice as large, and this goes for the entire range of stimulus voltages used. So basically, in this transgenic mice, which blocks where where vesicle release from astrocytes is blocked, we see that synaptic transmission from CA3 to 3A1 neurons is about twice as large. So the investigators went on to figure out what in fact was happening, and what they discovered is that normally there is a tonic release of ATP from astrocytes. As you recall from 2 slides ago, we showed that ATP can be released from Muir glial cells in the retina when they're stimulated. Well, what was found in these experiments is that ATP is tonically being released from astrocytes. This ATP is converted to adenosine, as it was in the retina, and activates, in this case, A1 adenosine receptors on presynaptic terminals of the CA3 axons. And the effect of activating A1 receptors on presynaptic terminals is to reduce the strength of synaptic transmission. So Normally, in the wild-type mouse, astrocytes are releasing ATP, leading to adenosine inhibition of synaptic transmission. And when the ATP release is blocked, as it is in the transgenic DN snare mouse, then this tonic inhibition of synaptic transmission is removed, and the amplitude of synaptic transmission is increased. So, the bottom line is that in the brain, normally, ATP is being released from astrocytes, which is reducing the strength of synaptic transmission. So this ATP release is directly controlling the strength of synaptic transmission, and when it's removed, the strength of the synaptic potentials grows, in this case, twofold larger. The investigators in this experiment went on to demonstrate that this ATP release from astrocytes not only modulates the strength of synaptic transmission, but can also influence synaptic plasticity. So what they did was to investigate LTP, long-term potentiation, at the CA3 to CA1 synapse. And if we first look at the wild-type animal, here is a measure of LTP where the strength of synaptic transmission is monitored before tetanus. At time equals 0, the Schaefer collaterals are stimulated at high frequency, and then this leads to a potentiation of synaptic transmission, which is shown here in the filled circles, and this decays, but we still have a potentiated, after 1 hour, we still have a potentiated synaptic response that's more than twofold the pre-tetanus amplitude. So here is the typical LTP that's seen in the wild-type animal. Now, when the same experiment is done in the transgenic animal, where exocytosis is blocked by the DN snare, the dominant negative snare, we see that LTP still occurs, but the magnitude of LTP is significantly reduced. And the reason for that is the following. Remember, in the DN snare animal, the synaptic responses were larger to begin with. And this, in turn, affects the amount by which the synaptic transmission can be potentiated. If you start from a larger synaptic response, you have less room to increase the response amplitude before you reach a ceiling level. And that is illustrated right here. So these experiments show that release of a gliotransmitter, ATP, from astrocytes not only modulates synaptic transmission, but actually modulates the strength or magnitude of long-term potentiation. And this in turn implies that release of gliotransmitters from astrocytes can control the mechanism that is thought to be responsible for memory, and thus glial cells are indeed playing a very important role in basic mechanisms of learning and memory. We've seen in the previous 3 examples that release of gliotransmitters from astrocytes can modulate neuronal excitability and synaptic transmission. Now, this does not occur by direct synaptic contacts between glial cells and neurons. Remember, at the tripartite synapse, the glial cell processes are surrounding the synaptic cleft of the neuronal synapse, but the astrocyte does not form a true synapse directly onto the neuron. Instead, the gliotransmitters are released into extrasynaptic space, and they diffuse 2 extrasynaptic receptors on either the pre or post synaptic membrane. It turns out, and this was a discovery made relatively recently, that there is one type of glial cell that does form direct synapses, and in fact both excitatory and inhibitory neurons can directly synapse onto this type of glial cell. And this is an NG2 type of glial cell, the oligodendrocyte progenitor cell. And in these experiments, it was shown that if neurons are activated, a synaptic potential and synaptic currents can be recorded directly from the glial cell. This is an electron micrograph showing a presynaptic terminal of a neuron with the synaptic vesicles, and this dark profile here is the process of a particular type of glial cell, this NG2 cell, which is distinct from astrocytes. And if one records from this NG2 cell and activates the neuron, one records true synaptic currents and potential. So here is a record from the NG2 glial cell. Under voltage clamp conditions, one records an inward current, and then if one records under current clamp, one sees an EPSP, an excitatory postsynaptic potential. In this case, the synaptic transmission was caused by release of glutamate from the neuron and activation of AMPA receptors on the glial cell, on the NG2 cell. It was subsequently shown that there are also GABAergic synapses between neurons and NG2 cells. In the case of the NG2 cell, release of GABA and activation of GABA-A receptors leads to a depolarization of the glial cell, not a hyperpolarization. It's also been shown that these synapses between neurons and NG2 cells occur in many regions of the nervous system, including the cerebellum, the hippocampus, the cortex, and the corpus callosum, among other areas. We have seen in the previous examples that signaling can occur both from neurons to glial cells and from glial cells to neurons. When neurons release neurotransmitters, these can activate receptors on astrocytes, leading to calcium increases. In turn, when calcium increases occur in astrocytes, this leads to the release of gliotransmitters, which can modulate neuronal excitability and synaptic transmission in neurons. It turns out that signaling can also occur from one glial cell to another. Now, astrocytes are coupled together by gap junctions, and this coupling forms a large network or syncytium of astrocytes that are functionally connected together. When a single astrocyte is stimulated, such as with a neurotransmitter, causing a calcium increase in that astrocyte, this localized calcium increase can spread out to other astrocytes in this coupled syncytium, leading to a propagated calcium wave that travels through the syncytium or network. Now, there are two mechanisms by which this calcium signaling is propagated from one astrocyte to another. The first mechanism involves the diffusion of IP3 in the intracellular messenger, IP3, from one astrocyte to another via gap junctions. So if we go back to the initial activation of a single astrocyte, typically when a neurotransmitter is released, this will activate metabotropic receptors on the astrocyte, leading to the generation of IP3 in that astrocyte. The IP3 will then open IP3 receptors on the endoplasmic reticulum of the astrocyte, leading to release of calcium from the ER. This is what generates the calcium signal in the astrocyte. Now, the IP3 that's generated can also diffuse to other astrocytes via gap junctions, and with the diffusion of the IP3, this leads to the release of calcium from the endoplasmic reticulum of these other astrocytes. That's the first mechanism by which glial cells can signal each other. The second involves release of ATP. When calcium increases in a single astrocyte, as we've already seen, this can lead to the release of ATP. Now, there are ATP receptors, purinergic receptors, on the surface of astrocytes, and when these are activated, this leads to production of IP3 and the release of calcium from internal stores as well. So, when the initial astrocyte is activated, this results in ATP release. The ATP can diffuse to neighboring astrocytes, leading to IP3 production and to release of calcium from the ER. Together, these two mechanisms generate these intercellular calcium waves that propagate out spherically as the calcium signaling spreads out through the astrocyte network. So what could be the purpose of these calcium waves and signaling between glial cells? Well, this may be one way in which glial cells can influence distant neurons or in fact, distant blood vessels. So if neuronal activity causes a local increase in calcium in a single astrocyte, if this calcium increase propagates to other astrocytes, this could lead to the release of gliotransmitters from distant astrocytes and to the modulation of synaptic transmission. In addition, as we will see a little later in the lecture, calcium increases in astrocytes can also lead to the release of vasoactive agents onto blood vessels, leading to changes in vessel diameter. So in the same way, a calcium wave could result in the regulation of blood flow in a vessel that's somewhat distant from the neuronal activity that initiated the calcium increase to begin with. Now, the initial experiments that demonstrated the presence of calcium waves in astrocytes were done in ex vivo preparations, either initially in cultured astrocytes, then in brain slices, and in whole mount retinal tissue. So the question arises as to whether calcium waves occur in vivo. And indeed, both in the retina and the cerebellum, it has been demonstrated that calcium waves occur spontaneously in the in vivo animal. And in addition, it's been shown that in the cortex of Alzheimer's mice, calcium waves are spontaneously generated, and the site of initiation of these waves occurs near deposits of beta amyloid plaques. So that raises the intriguing possibility that calcium signaling and propagated calcium waves could be involved in some way with the pathological processes occurring in Alzheimer's and perhaps in other diseases as well.